moving. So last time we've been working with swarm intelligence, and as sort of a subset of distributed AI. And last time we were focusing on, and I will post these notes as usual, but last time we focused on ant colony optimization. And I specifically focused on a simpler version of ant colony optimization known as ant system, which was kind of the original ACO. So this is usually goes by ACO, and the original one was this ant system, which was ant system was credited to Marco Dorico primarily. And ant system followed kind of the structure that I talked about last time, but the and I'll kind of rehash some of that here just as a refresher, but it didn't have the performance as a lot of other off-the-shelf solutions for traveling salesmen, et cetera. And so ant colony optimization, the big difference there was that they added in a lot of heuristics that would allow the ants to perform better, so find solutions better for it. So as an example, they would add a form of elitism. So if an ant found a particularly good path, then rather than getting back to the nest and then choosing a possibly totally new path based on the pheromones, it might remember its old path. In other words, it kind of could ignore the other pheromones while continuing to lay its own pheromones. And so there are all of these little additional heuristics added to ant system that were built into ant colony optimization that tried to improve its performance. But the essence of it really goes back to this original ant system. And the basic idea there was trying to leverage this thing called stigmergy for communication. And so rather than having direct peer-to-peer communication between individuals in this simulated system, then each individual would modify the environment in a way that would coordinate with other individuals. And so I would sort of define stigmergy as coordination by means of persistent, and the key there is persistent, modification of the environment. And ACO was specific for these combinatorial problems where every decision vector came from a discrete space. And in essence, you basically pictured an ant that was at some nest, and it then would then walk through the different variables. So these are decision variables. And each decision variable has a level because it's discrete. And so at, you can imagine if X1 is here, X1 might have different options for X1. And you could imagine each of the different dimensions of the decision variable would each have their own different levels. And there you could picture some food source. And so the idea would be that an ant, initially you would draw a random decision vector, which would be kind of like connecting a bunch of random levels here. And you get back to the food, and the ant somehow remembers the path back on its own. And as it comes back, it lays a pheromone based on quality of solution. And then over time, those pheromones get deposited on these different areas. And so you get pheromone that gets deposited on these. And those pheromones shape the distribution of how the next ant chooses its particular levels in this decision space. And so you're really generating probability mass functions at each particular section here. So you can imagine there's a probability mass function which guides the selection of each one of these levels. And we mentioned that continuous versions of ACO have cropped up, and in the continuous versions, they basically convert. So if I wanted to convert this continuous from a probability mass function to a probability density function. And so the idea here would be that if you have a continuous choice for each one of your X1, X2, X3, and so on, then you might start out with a uniform distribution. You lay down pheromone, and those pheromones gradually prop up certain parts of the distribution and maybe push down the rest of them. And so you could imagine extending this from the discrete case to the continuous case, but originally it was envisioned just for this discrete case. And you could also imagine taking a continuous distribution and then chopping it up into finer and finer levels and making it sort of a probability mass function over more and more outcomes. And that was the essence of ant colony optimization. So this idea that we are trying multiple solutions initially and then using their relative success to then reshape the population of solutions. And so I hope you can kind of see that there is some aspect of evolutionary computation in this, in that every generation, you end up reshaping the past generation based on the past success. And so there is definitely a relationship there, although it's not implemented like a genetic algorithm. So before we move on with those things in mind, are there any questions about ant colony optimization? All right. So ACO is pretty popular. You can find it, it's pretty much anywhere where you can find an off-the-shelf tool that provides you genetic algorithms, you'll probably also find ACO. Okay, so what I want to move on today is to talk about two other examples of this kind of swarm intelligence. And the next one is this bacterial foraging optimization. So I'll just put that up top here, bacterial foraging optimization. And this is actually an approach that my advisor, Kevin Passano, published in an IEEE journal from Control Systems. And that I've published, or put, I think, that article online, as well as a chapter that he wrote about this one and how it relates to some others in there. And the idea here, this is motivated by the behavior of these bacteria. And so bacteria have been associated with a particular chemotaxis strategy. So I'm going to write this. So chemotaxis. So this is a taxis represents how an animal moves through a space, and a chemotaxis represents how animals respond and move according to chemical cues throughout the space. And so bacteria have this interesting behavior of the so-called run and tumble. And so the idea is that if you have a flagellated bacteria, so you have this sort of long bacteria, and it has all of these flagella coming out the back of it. The way these flagella on these particular flagellated bacteria are put together, if they rotate in one direction, they create a consistent movement and causing the bacteria to move. So in the run mode, you get a bacteria that will end up moving consistently in one direction. And that comes from the flagella rotating. And I always forget which one's which, but let's say they rotate counterclockwise or anti-clockwise. And that creates consistent agreement in the flagella, and that ends up creating this movement in one direction. Now, what's interesting is that periodically the bacteria will start rotating their flagella in the opposite direction. And when you do that, it causes the flagella to kind of get gunked up with each other, and the bacteria just starts to spin. And so the so-called tumble is when the flagella rotate clockwise. And again, I could be wrong on which one's clockwise and which one's counterclockwise. And this creates sort of a disagreement that causes the bacteria to just move in kind of a random direction. And so you get a random reorientation. And as people started to study this, what they learned was that bacteria would tend to run up until a point at which they reached a decrease in a chemical concentration gradient. And so they would run while the gradient was increasing, but at the instant they hit a decrease in that chemical concentration gradient, they would tumble. And so, so basically the process was run while moving up chemical gradients, or nutrient, I'll say nutrient gradients, and then tumble at a decrease in nutrient. And so this together, if you combine these two, you end up actually getting an interesting and effective search strategy for an individual bacteria to climb a nutrient gradient. And so, and if you can implement this, this run and tumble technique, and it does work pretty well for an individual bacteria. The downside of this is it's very easy for it to get caught in local traps. And so this is, I guess I could say this is effective in moving uphill, but gets caught in local traps. And so the thought is, how, could bacteria use social information to try to get the best of both worlds? You can imagine a bunch of bacteria climbing independently, and as those bacteria climb independently, some of them get stuck and some of them don't. But if they have some way to advertise that they're in, that one of them is in a better spot than another, then maybe that one can ignore its local nutrient gradient and start running and tumbling toward the bacteria at a different spot, and maybe get out of their local trap, and maybe they can all aggregate together in a particularly rich spot. And so even bacteria tend to have sort of a different relationship to relatedness than a lot of other organisms, because they can do horizontal gene transfer and all these things. But in general, there are actually a bunch of benefits that bacteria get from being together. And so, there's might be other reasons why you might be not only want to be at the top of your nutrient gradient, but be at the top of your nutrient gradient with other bacteria. And so it was found then that bacteria are not only responding to nutrient gradients around them, but they are emitting chemicals around them into the space around them and responding to these social chemicals. So bacteria not only pay attention to the external environment, but they create a social environment that they also pay attention to, and that does seem to drive their bacteria, or drive their behavior. And so when we think about the function, so you could say, what is the optimization objective for a single bacteria, then we can break it into two parts and evaluate it at a single, so this is, I'll say, evaluated at a point in space. I would say a single point in space. And so there is a component J, which is basically the nutrient concentration, which from an algorithmic point of view, this is where we put our optimization objective, the thing that we want to minimize or maximize. And oh, I'm sorry, I see now people are saying they can't see what I'm writing, that I've already moved forward. Okay, well, let's see if I can reshare this screen and to fix that. All right, I'm working on reconnecting. Like I said, it takes a couple of times for the screen to reconnect when we do it this way. This worked well earlier in the day and is not behaving as well as we'd hope now. So let me try one more time and then I'll go to an alternative solution. Share. Mm-hmm. All right, so I'm going to try again connecting over Zoom over my tablet. So I'm going to try muting my audio on the other machine that I'm on. All right, can people still hear me? I understand that you can't see a screen yet. Great, thanks for that. All right. So, and now can everyone see the screen? Great, okay. So this was our first page, which I think everybody saw. And I think I've tricked it into not having feedback. So hopefully everybody can see everything and also saw that transition to this second page where I've got this bacterial foraging optimization. And again, sorry that you didn't see this. So I was just sort of drawing this, other bacteria up here. This is the body of the bacteria, and these are the flagella back here. And the bacteria has two modes of behavior, the run and the tumble, which is what we were talking about here, the run and the tumble. And that's what generates the back and forth that allows an individual bacteria to move effectively through nutrient gradients, but gets caught in local traps. And so it turns out the bacteria also emit chemicals and they respond to the chemicals emitted by those other bacteria. And so effectively, although we think of the bacteria group, oh, I see, the nutrients are from the environment outside the bacteria, not internal the bacteria. That's right, the nutrients, we're thinking of, you just inoculated a plate with bacteria, and that plate of bacteria has a bunch of nutrients on it. You can think of them as food for the bacteria, and the bacteria wants to go to the location with the most food. And so those are the nutrients, and they're not internal bacteria. The bacteria would like to internalize them. Great. And so the idea is that we are going to take those nutrients, so that's what we have down here, and we are going to model our optimization objective as a fictitious nutrient concentration for our simulated bacteria. And that's what I'm calling this function J. So that is our optimization objective. But the bacteria are going to see not only J, but a JCC, which is, I'm saying this is our cell-to-cell attraction. So, so I'll just, maybe I'll put this down here. This is cell-to-cell attractance. And we'll see that this also might involve repulsion. And so this together is the kind of individual level concentration guiding run and tumble. So it's almost as if the bacteria do pay attention to the outside world, but they also are focused very much on their social environment. And that's how they decide where to move. And so what does this JCC look like that we've kind of added onto our optimization objective? And it typically is going to be something like so. So we're just going to define it as, it's my triangle equals is defined as, we're going to say it's the sum of so we'll say this is the, how do I want it to, maybe I'll just simplify this. So let's imagine that we've got, and the goal here, our goal, by the way, is minimization. I know the bacteria, we want to say that they're climbing uphill, but just for just because it's conventionally optimization literature to minimize, then we're going to say this is minimization. And so our cell-to-cell attraction is going to be some constant that I call D attract times an exponential function, which you can kind of think of as a kind of logistic regression. So there's going to be some weighting in here, so I'll also call it minus W attract, that then sums over the positions of all the other bacteria in the population. So I've got a population P, and let's say I've got a bacteria that is got a position so we can think of theta M as maybe the current position of our bacteria, and theta M to the I, and that's not meant to be an exponent, that's just another identifier, as is the position of another bacteria in, so well, the M, I guess the end, I don't know why I did M to the one here. Yeah, I'm not sure why I've done, so that the reason this I is here is that I originally was going to say there can be multiple attractants, and you can imagine, you know, emitting different types of attractants, and this is one type of attractant, but I got rid of that sum because I was just trying to keep it a little simpler. But that, so that I in there is supposed to be kind of the amount of attractant emitted by a particular bacteria. So you can think of this, this theta M here as, maybe I didn't mean to maybe sub M, but this is supposed to be sub, we'll say, I'll just say J. And so this is maybe position of this bacterium, and this is position of other bacteria. And so the idea here is that this attractant chemical is able to penalize positions in this exponentially weighted way. So you've got this, every bacteria has got this sort of penalty function, which is quadratic. It gets scaled by this attraction scalar, and that whole thing gets weighted by some exponential. The idea being that if they're very, very far away, then this attractant signal goes away. And if they're very close, then it's as strong as the coefficients allow it to be. And then you would have some repulsion signal, which will take the same form. So except everything will be a positive. And so if I have some H repellent, and it has some exponential weighting as well, and then we sum over our whole bacteria. And again, we've got whatever our current theta is minus the, so I could say this is maybe JCC, I will say on bacteria J. And then this is again has our M squared, and I'll put the little I in there just to be consistent with what's above. And so now we have the ability, I see here, so what, can I reiterate what is the input to this optimization function? So my decision space is the, so my decision space is mapped to the positions of the bacteria. And so, and the this I mentioned before, we can probably erase I. If you look in the full version of this bacterial foraging optimization, then there are multiple attractants that you can put in here. So there's a different summation that I've excluded to try to make things simpler, that in my original notes I included, but I didn't want to throw all of that in here. So for now, we can just, I'll just etch out, I'll say, let's not worry about I. And to capture the basic idea here is that we just are penalizing, we are either amplifying or penalizing our distances to and from different bacteria. And so you can imagine bacteria are attracted and then D attract and W attract, these are just constants. These are just coefficients. So these things here, these here are coefficients, hyperparameters. So I'll mention that. So you set how attractive you want each bacteria to be and how repulsive you want each bacteria to be, and the weight, and you also set how attractive and repulsive you want the relative distances in space to be. And so that's what the outer, the D attract and H repellent represent, whether you want attraction or repulsion in general. And then inside the exponential, the W attract and W repellent are the weights in which you put on distances between bacteria. So you can say, how close do you want bacteria to be? And if they get too close, then you want them to sort of be, to push away very quickly. But if they're very far away from each other, how do you want that balance to be? And so those weights allow you to do that. And so I'm looking down through questions here. So the input to this optimization function, again, are the positions of the bacteria. So you can imagine each position is a different decision vector. And so as they move around the vector space, then they're moving to different potential solutions to your optimization problem. D and H are hyperparameters, we got that. And then at any iteration step, will big O be P squared? Is that right? Well, I guess you'd say that if we're talking about with respect to the number of bacteria, I guess you do have to calculate all of these different things. And so there will be a trade-off between the number of bacteria searching and the calculation of this, sure. But I'm not going to talk specifically about the complexity of these things. Usually we're talking about like, this is similar to genetic algorithms. Maybe there might be 100 individuals or something like that. So hopefully this is not too difficult to calculate. So that's the basic idea of how each individual responds. And so each individual is going to run in Tumble. So basically what you're going to do inside an algorithm like this is you generate, for every bacteria, you remember the previous value of J plus JCC, and then you end up doing a run so you can move in a random direction. And if the new value after you move in that random direction has gone, in this case, up because we're doing minimization, then you do choose another random direction and run in that direction instead until you find an upward direction. So that's the basic idea at the individual level. And so you might want to ask though, like, if I have a very large decision variable space, so if I have, so each bacteria is moving around in, in a 100-dimensional space, how do I choose a random direction? So if I'm moving in a 2-dimensional space, it's easy for me to choose a random angle, but how do I choose a random angle in a 100-dimensional space? And so I'll mention kind of as an aside is I could say random direction in high dimensional space. Then all you have to do is draw basically a vector. So we'll say draw a vector that I'll call it big delta, that comes from the space of -1 to 1 for however, this is how many, the dimensions of your space. And then just normalize that vector. And so your random direction is then just whatever that vector is divided by its norm. And then this right here is guaranteed to be a unit vector in a random direction. And so with that, you could imagine building a relatively simple motion rule where you use this to implement your tumble. And you could come up with another hyperparameter to determine how long you want to run. So you run for a certain amount of time. If that didn't work out very well, then draw this big D delta, normalize it, turn in that direction and add that to your current position, and then move in that direction for a certain amount of time, reevaluate, and so on and so forth. And so that's kind of how we implement tumbling. The ultimate goal is each bacteria wants to, in this case, minimize, in reality, maximize its gradient, but you can think, or maximize its nutrient concentration wherever it wants to maximize the location, find the location of the maximal nutrient concentration. Our goal is usually to minimize a function, so we kind of change the direction of everything so it's minimizing, but it's just like in the genetic algorithm. I'm looking to build a population of bacteria that collect around the global optimum defined within this position space. And so hopefully all bacteria find this, but it's just like the genetic algorithm. You might end up getting these separated populations if their attraction and repulsion regions are such that they don't have a lot of coupling. And that might be okay for you, but this algorithm tends to generate groups of bacteria that move together into regions that are better and better until hopefully the group of them finds the global optimum. How do we still ensure that for how long we are going to randomly look for a direction in which the situation is better in terms of J or JCC? Yeah, you can see details of how this particular algorithm is implemented in the paper that I posted online. But the basic idea is that the randomness, it allows you to search the space, and whenever you end up going in the wrong direction, you basically reject that. So this is really just coming up with a proposal direction. So you can imagine a bacteria kind of tests the waters and sees if it should move in a particular direction. And if that direction is moving in the wrong direction, then it randomly turns and moves in a different direction until it only is sort of okay to keep running if it has moved in the right direction. And so if you do a run and you evaluated you have done better than the last time you did the run, then you continue to do a run in that direction. And you only have to generate a random direction when you've gone in when you found that your run has taken you into a worse spot of the space. And so you don't ever, the termination conditions generally are set by the outside designer of the algorithm. And maybe it'll just be the number of times through the algorithm. Maybe it'll be some convergence conditions. Again, that's up to you. But as far as the bacteria is going to view, it's just going to keep trying to move in random directions so long as it moves in the wrong direction in the nutrient space. I don't see how the unit helped with the problem of having 100 dimensions. It's still a unit vector has some, well, what I meant by the unit vector here is if I only had 2 dimensions, so I have like x1 and x2, if I wanted to define a random direction to move in, let's say I'm in here in the space, then I know that I could just draw a random angle. So a random angle from the space of say 0 to 360 degrees, in which case I can just choose whatever that random angle is and move in that direction to that new point. But the issue is, how do you draw a random angle when you have a wide number of dimensions? And so I'm going to sort of, an abusive notation here, draw these multiple dimensions. We can't just draw a single scalar to tell us which direction to move in. And so the way you draw a random direction in high dimensional space is just by drawing a random vector that has from that space, and then normalizing that vector, and then that will give you a unit vector in the high dimensional space, which is equivalent to effectively drawing a random direction. So there are a bunch of other ways you can imagine drawing random directions. Like you can imagine, oh, I'm going to draw a random direction in one dimension, and then a random direction in a different dimension, a random direction in a different dimension. And this is a way of drawing a random direction in all of the dimensions at once, rather than having to hop through one at a time. Okay, good, great. All right, so this is what the individual bacteria do. Now, what's interesting about this bacterial foraging optimization is not only do you have this kind of swarm intelligence where the individuals are socially guiding themselves to different regions in space, but there's an evolutionary component as well. And so because we've got these individual bacteria and they're all sitting at different nutrient concentrations, at the end of every round, after you've done this movement rule in every bacteria, you can decide to kill certain bacteria, eliminate them, and then redisperse them to other parts of the space. And so there is a bit of an evolutionary component in that you can come up with a rule to eliminate poor performing bacteria, and then regenerate new bacteria as you wish. So it might be that you're going to regenerate them close to the ones that did very well, like a clonal reproduction. Maybe you're going to regenerate them randomly. I think you can see there's a bunch of different opportunities here, but this framework of bacterial foraging optimization was developed in order to try to blend a bunch of different components of swarm intelligence and evolutionary algorithms together into one. So you've got this kind of this thing where they've got a chemical concentration that they send out. It's very kind of pheromone-like, and so they can use that pheromone to drive them toward each other. You've also got this evolutionary component where you can penalize some more than others. And so that's kind of, again, got this sort of evolution thing here. There's a bunch of degrees of freedom that you can play with, but that's kind of it's sort of meant to be a tool on which you can bring all of these things together and work in a continuous optimization space. And so that's bacterial foraging optimization. Now, one thing that I do want to mention that I think is kind of interesting about bacteria in general is that when people realized that bacteria did run and tumble that was this way, they initially thought that a bacteria might be large enough to be able to sense a nutrient gradient in one place. In other words, you could say, what is the nutrient gradient at my head? What is the nutrient gradient at my butt? If I measure the difference between the two of them, then I generally know what the nutrient gradient is across my body. Sorry, what's the concentration at the head? What's the concentration at the butt? Then you could calculate an approximation of the nutrient gradient along the body of the bacteria. And maybe that's a way to estimate the gradient. But it turns out that when people looked more closely at the scale of how these nutrients changed, that the bacteria were actually too small to get any useful gradient information from sitting in a single space. It's so they kind of had the vanishing gradient problem that you see in training neural networks, is that to a bacteria, if you only measure at one particular spot, you always see basically no gradient. So what that revealed to people is that bacteria, as small as they are and as dumb as you might think they might be, have some form of memory that we still don't quite understand. So as bacteria move around, they're actually sequentially sampling their environment and using that to figure out whether they've moved up or down gradients. And it's still very mysterious and kind of very interesting and exciting, but at least shows you that you can have memory in natural systems in places that you may not have even expected. So not only are they sending out chemical social stuff, but they also are storing memory inside. The question is, how is repelling helpful? So again, so this bacterial foraging optimization was meant to sort of blend a lot of the things that we've already seen in like genetic algorithms. And so in genetic algorithms, we've seen in niching methods, you try to promote diversity. And so you might not want a bunch of bacteria all exploring the same part of the space. And so if you have a reason to believe that once you're at a particular decision vector, there's no value in looking very close to that vector, and so basically you should be searching out farther from that, then you can crank up the repulsion part of this, and that creates a bubble around these bacteria that prevents them from aggregating too tightly. So whether repulsion is good or not is going to depend on the type of optimization objective, but I would draw a relationship to the diversity preservation mechanisms in genetic algorithms. Does that help? Great. All right. So that's bacterial foraging optimization, or at least bacterial foraging. Okay, we've got, in the case of bacterial sampling, how do researchers make them concentrate on a point and conclude they can't make sense when the input they get is all one point? How do we get their memory? So the physics of looking at the nutrient gradients that bacteria were effective on climbing of were such that that there was no sufficient signal. So at the scale of these bacteria, the nutrient gradient, you could say the gradients that the bacteria were climbing were of a length scale that was much larger than the bacteria themselves. And so because of that, then the gradients that it's kind of like if any of you are familiar with numerical integration, there are so-called things as stiff differential equations. And if you build a numerical solver that needs to solve a stiff differential equation, that basically means there's tiny variations you need to ignore. And you should only pay attention to the large variations in the differential equation. And it's kind of similar here with the bacteria, is that the gradients the bacteria were climbing were too slowly changing for the bacteria to be able to get any useful information at the length scale of a particular bacteria, which has led bacterial scientists, people who study this sort of thing, to conclude that the bacteria must be sampling from their environment over time, as opposed to just using a local rule based on what they can see at the current instant of time. It's still kind of mysterious on how they do that, but that's the current understanding, is that they most likely are somehow remembering a previous nutrient concentration and comparing it to the current nutrient concentration to figure out if they've gone up or down. Okay, great. All right, so that's bacterial foraging optimization. I want to then pivot to something that, so bacterial foraging optimization is meant to be kind of a complex everything of metaheuristics. You know, it's got evolutionary stuff, it's got social stuff, it's swarm intelligence, optimization, it's continuous, it's potentially could be discrete, and but it's kind of ugly to put together. And so, so I'm going to flip now to particle swarm optimization, which was meant to be a simpler approach that has similar features, but maybe with less of the evolution, and actually on purpose, less of the evolution. But before I switch to PSO, are there any other questions about this basic idea of bacterial foraging optimization? And the things I'm trying to get you to carry away from it is augmenting an optimization objective with these ideas of repulsion and attraction in order to guide the distribution of a group through an optimization space in a run and tumble like way. So each individual has a way to climb locally, and then but the nutrient, the gradients that the individuals see are related not only to the thing we care about, but are also related to a social pressure that helps them attract towards each other and away from each other. So if there are no questions about that, then let's flip to particle swarm optimization. All right, so the last one of these swarm intelligence that I want to talk about is PSO, or particle swarm optimization. And so some of you who work in machine learning might be familiar with particle filtering. Particle filtering uses particles in kind of a similar way, but particle filtering is sort of only tangentially related, because they're using an ensemble of particles to make guesses at things. Can we point to more material later, which gives more light on how problems were modeled for bacterial swarm optimization? So if you go onto Canvas into this unit, I have a couple of different references on bacterial foraging optimization that you can take a look at. The article, which is, I think it's like the Pasno, I forget which, 2002, maybe 2005, was an IEEE, I think it was a Control Systems Magazine article. And so it's meant to be kind of a nice overview of possible applications. And then it even shows examples of how it's used and what the performance is. And so I definitely recommend you take a look at those references online. And then if you have questions about that, get back to me with questions, and I can try to answer those or find more references for you. All right, great. So particle swarm optimization, this is, so we're in thinking about swarms solving problems. We're in the kind of thinking about, so particle swarm optimization is older than bacterial foraging optimization, but it is still motivated by this idea of having attractants, having things that attract and maybe things that repel. But the question is, what was the motivation for going in this direction? And the idea is let's say you're optimizing a neural network. So let's say you're trying to train a neural network, and we'll talk a lot more about neural networks specifically later. And in a neural network, you have all of these different weights that need to be set. And these are your parameters to optimize over. Now, the problem with neural networks is that for a given loss function, so for a given objective that you're trying to solve with your neural network, so you're trying to build a classifier or something along these lines, that there often are a wide variety of very similar choices of weights that give you similar performance. So you can say that you can have similar loss values, and so we like to minimize loss across wildly different weights, weight combinations, I guess I could say. And so this creates a problem for genetic algorithms. In theory, so the idea would be here is that if your genotype are these weights, then what you're saying here is that there are a bunch of different ways to instantiate a set of genes that are all going to be performing equally. And so it's going to be difficult to say which one's better, and you're going to be fighting among these different genotypes rather than exploring one particular genotype and kind of doing the best you can there. So if you basically say that I'm not as worried about finding the global optimal, the best weights, but I need to find some pretty darn good weights. I don't want to waste a lot of time investigating different sets of very similar weights that are very far away from each other. It would be better for me to kind of cluster around a set of decision variable candidates that are similar, but we can tune. So if you want to tune in an area and you're less worried about finding the absolute best area among a bunch of very similar areas, then is there a way we can build an algorithm to do that? And PSO was meant to do that. So like the motivation behind PSO is to build a population that clusters together around good solutions. So we're really biasing things more towards exploitation than exploration. And so we want the PSO to do a good job searching, but we want to kind of keep everybody from drifting too far away from each other. And so instead, we're going to try to have them maintain some cohesion and then have that population move hopefully together and drift towards better and better solutions as a group. So we're trying to get the group to move like a blob towards better and better solutions, and then have members of the blob optimize within those solutions. So that's kind of our motivation behind PSO. And so this is motivated by there are these old models that come from the computer graphics community. So there's, you can go back even farther than this, but this is the reference that most people say was kind of most motivational, and then all of the other references that predated these weren't quite as popular. And so post hoc, people point back and say, well, actually you saw people doing the exact same thing in literature that's 10 years older than this, but still this kind of kicked things off. So there was a guy named Craig Reynolds, and he had these things. He was interested in building for computer graphics things that would generate large groups of animals or other bird-like or flock-like objects that would move in a realistic way in a computer animation. And so he called these things boids, where a boid is a bird-oid object, where oid here is kind of, basically like. And so these are bird-like objects. So he was trying to build computer graphic animations that looked like realistic versions of flocks of starlings moving around. And this has been applied to terrestrial animals as well. If you've ever seen Disney animations of wildebeest running across, some African landscape, then they probably used something that was derived from these original Boyd's models from Craig Reynolds. And the basic earliest versions of this, like I said, I guess I should really tip my hat to there are people like Aoki from 1982. There are several other articles from the 70s even, but Aoki kind of did this from actually studying fish population and how fish moved around. But Reynolds came along kind of, as far as I can tell, ignorant of these things, just because, it was hard to figure out, back then we didn't have Google Scholar. And so this, the Boyd stuff came out in 1987. And again, that's kind of what's gotten the most popular in this community. And the idea is, what if you have an agent this, this triangle agent here, so is moving in this direction, and it has some perceptual radius around itself. And you can think of it has some distance that it can see. And it has some, some angle. So it can see this angle around itself, and it can see out this far. And so it then makes movement decisions based on what it can see. And so for all agents, in this region, we want to separate to avoid collision. We want to align to a common heading. We'll say, this could be an average heading. And we want cohesion toward an average position. And you can implement these 3 rules on every single agent, throw all the agents, and you can basically put these into a weighted combination. And so you can see how this is the bacterial foraging model kind of borrowed a lot from this idea as well. And then you can also do things like add obstacle avoidance. And that sort of helps you maintain feasibility. So you don't drive off the end of, in our case, a decision space, but in Reynolds' case, he was trying to make sure the wildebeest didn't run off the side of a cliff or something like that. So you could add kind of fake agents that stay in place that are highly repulsive, and you could put that in here. But in sort of an unconstrained area, these are the 3 rules that kind of deal with everything here. And so you could imagine also adding, so these rules alone, then you can get some pretty cool looking behaviors, but they're not like moving any in the directed way. And so you could also add to this a weak attraction to an environmental stimulus. And this is how you could get some gradient following. And when we do that, then I think you can see then there's a very close connection between this and the bacterial foraging optimization. You've got the kind of cell to cell. So this is similar to that JCC that we saw before. And then over here, this is similar to that J. And they're following both of those simultaneously. And then you can get some cool movements where everybody's sort of trying to stay close to each other, but not too close. And then the group is hopefully following some gradient as they may be. And if you think in terms of computer graphics, maybe they're trying to always run downhill or maybe they're trying to run uphill or something along those lines. So there was the Boyd's model that was very influential. There was also from the physics community, when we see how do we encode them. So this, again, I'm sort of just thinking in the computer graphics. So this is, I think was in ACM SIGGRAPH, by the way. 1986 is where we see this, where Boyd's were introduced. And so from the computer graphics perspective, they weren't interested. They were just said, well, these are entities in a 3-dimensional space like birds, and we will render these birds, but we need to figure out the position of every center of each one of these birds in between renderings. And that's what he was trying to say, like, where do we put the birds from time step to time step? What we would do is, if we want to use this as an optimization algorithm, we would do the same thing we did with the bacteria, where the positions in space are decision variables. So each position in space could be a vector of weights, and those weights could correspond to weights of a neural network. And this group would then swim around weights that are very similar to each other, but hopefully the cluster could move through large regions of space, allowing them to search, but then they never kind of break up their search. They kind of keep their search together at all times, kind of like, again, a flock of starlings. How do we decide stimulus if, in the context of neural text, how do we decide stimulus if all we use is this idea here? Well, so the stimulus in a neural network case would be that loss function. And so everyone's trying to move down the loss function. They're trying to make the neural network a better and better approximator of a function. But then meanwhile, all of the different candidates, neural networks, are trying to keep their weights close together. So as we search in an area that's close together, but then hopefully that area moves to lower and lower loss functions over time. And how do we ensure there's cohesion? Well, that's the thing about these metaheuristics. We never can ensure there's cohesion. We just have to crank up the alignment and the cohesion so that we hopefully bring them together. And we understand that if we make these alignment and cohesion too weak, then the repulsion, the separation, may end up dominating and they'll split apart. And we'll have the same problem we'd have with your standard genetic algorithm. So in all of these metaheuristics, it's kind of more science than engineering, in that we set the parameters and study how they work, as opposed to getting a proof to guarantee that things will always stay together. Just like a flock of birds, sometimes it splits in two. Okay. Is there a specific literature that we can refer to know more on the math? And so, like I said, you can look up these ACM SIGGRAPH, Craig Reynolds papers. If you just do a Google Scholar for Boids, you'll find a whole bunch of stuff. And then there's also an influential paper by Thomas Vicek from 1995. This is et al. That if you search for, and I think it's probably a paper in, I almost positive this is going to be in Phys Review Letters, but I don't know off the top of my head. But in the Vicek model is kind of a mathematical instantiation of how you can make this thing work. You say there's a gathering around a good solution. I mean, various neural nets having different combination weights are gathering around. Yes, that's exactly right. So you can think of every bird in this flock being a different candidate neural network. And we want the neural networks to be close together. In that, we want the weights to be relatively close together so that we are sure that we're investigating kind of one area of the loss function, as opposed to trying to investigate a wide range of areas of the loss function all at once. So we have a limited number of neural networks that we're evaluating in parallel, and we are trying to evaluate them close together so that they can explore more deeply one region of space, rather than trying to get lucky and explore the whole space and not necessarily make a lot of progress. Okay. All right. So with the comment about the math stuff, and given that we've had these delays, it feels like maybe this is a good spot to pause and then pick up with the essence of, well, the Vicek model, which starts looking a lot like particle swarm optimization, and then pivoting directly to particle swarm optimization, which actually shows us how we implement this for optimization and the different variations of particle swarm, and so on and so forth. And so maybe we'll save the details of particle swarm for Wednesday, and we'll leave it for that. So what I'm going to do is, my office hours are at 4:30 in technically my office hour room. I'll stick around on this channel if there's any questions, immediately after class questions. But then between now and 4:30, I'll switch over to my office hours channel that I put on the Canvas page, and then I'll hang out there for about an hour if there's any other questions or concerns. But thanks for your patience today, and I'm gradually figuring out how to spin this things up faster and faster, and so hopefully on Wednesday it'll be a little more smooth start. So if you don't have any questions, you can feel free to leave the room. If you do have questions, then feel free to bring them up here, or wait and come back at 4:30 when I open up office hours. Thanks, everybody. There's a question about online lectures. It looks like we do have to switch to online for the whole semester. And so we're pretty much going to, I'm not going to make any major changes in the curriculum. Still going to deliver the lectures this way, gather questions this way. So I'm not trying to turn it into an online course. I'm just trying to provide an in-person course through an online modality. And if you're still looking for project partners, definitely post to the discussion boards, and I'll start forming the groups based on who's submitting things. And once I get a better idea of who's left without a partner, I can also start matchmaking. I don't have a tentative date, but I'll definitely provide people plenty of time, and you know, at least a week, if not 2 weeks, to do the. I mean, it's an online midterm. It'll be auto graded, open book. So I've got some of it written, but I just haven't, with the conversion of all the classes to online. I didn't get a chance to write it all up yet. I still have some homeworks that I'd like to put together as well. So I'm hoping soon, but I don't have a tentative date, and I'm sorry for that, but I'm doing my best. All right, I think things are quieting down here. So unless there's any other questions, then I think I'm going to leave this room. And if you need to come back for office hours, then join me at the office hours link, which you can find on Canvas or your email.